Hello everybody. Welcome once again to the last video presentation of Unit 14 Biomolecules. This is part 5. The last video we all had discussed about proteins, the structure of proteins, the applications of the structure of the proteins, and now we go towards the other unit or other component of the same chapter. We'll study about enzymes. Now, what are enzymes? All biological reactions are catalyzed by a special catalyst which is called as enzymes. Therefore, enzymes are all defined as biocatalysts which catalyze specific biochemical reactions. Now, when we are discussing about enzymes, enzymes itself are also proteins. So, we find almost all the enzymes to be globular proteins. We have discussed the last unit. What are the types of proteins like fibrous proteins and globular proteins? Globular proteins are those which are soluble in water, and enzymes are such class of proteins which are soluble in water. They are globular proteins. Now, enzymes facilitates a large variety of biochemical reactions, and what do? They act as biocatalysts in the sense they would be providing an alternate pathway with the lower energy of activation, and thus they would increase the rate of the reaction. Is that not the work done by any catalyst even in the industry or in the chemistry lab? Yes, the same thing is done by an enzyme inside our body. So they are very specific. The enzymes will actually catalyze a particular reaction, and it will be suitable for a suitable substrate only. Now, when we are talking about the specificity of the enzyme, let us take an example. The enzyme which catalyzes the hydrolysis of maltose to give glucose is called as maltase. Now, this is a biocatalyst, maltase, an enzyme which has a very low energy of activation, approximately about 6 kilojoule per mole, while the same reaction where you are trying to break down maltose in the presence of any other a suitable mineral acid or something, hydrolyze it to give rise to glucose, which is not catalyzed by our maltase, by any other catalyst in the laboratory. The reaction would have taken with an energy of activation approximately at round about 12 or 13 kilojoule per mole. So you are understanding that lower the energy of activation, faster is the rate of the reaction. So that is the function of enzyme. It lowers the energy of activation. Similarly, we have heard about sucrose getting hydrolyzed to give rise to glucose and fructose. If you use the enzyme sucrase, then the energy of activation required is just 2.1 kilojoule per mole. But if you don't go for the enzyme sucrase and just go for acid hydrolysis of sucrose, which you can do it in the laboratory at home, etc., it requires a high energy of activation, approximately 6.2 kilojoule per mole. So what does this tell us? It tells us that enzymes increase the rate of the reaction by lowering the energy of activation. So when we are listing the enzymes and giving them their names, the nomenclature is done by considering the utilization of the suffix ase. The suffix ase is added to the name of that particular enzyme, and how it is dependent upon the class of the compound or on the compound in which the enzyme is going to work. For example, you are having hydrolysis of starch, so you're using the enzyme called diastase. If you're hydrolyzing maltose, you're using the enzyme maltase. Here we are having the catalysis or enzyme catalysis of hydrogen peroxide going to be broken down to give rise to oxygen and water. The enzyme is called catalase. Pectin is going to be broken down to give rise to simple sugars, and the enzyme is called pectinase. If you're decomposing or breaking down a protein to give rise to smaller polypeptides, that time the enzyme is called pepsin. If you are breaking down the milk protein casein to give rise to curdled milk insoluble casein, and that the enzyme used is called renin. So these are some of the names given to the enzymes based upon the substrate on which the enzyme would act. Now, what are the major digestive enzymes? Because they play a dominating role in our digestion process. So, these are some of the enzymes where we are extensively having it utilized in our body. Carbohydrate digestion is carried out by enzyme called salivary amylase, which is produced by the salivary glands. And you know, the preliminary digestion process starts in our mouth itself, and it breaks down starch to give rise to maltose. That's what is being done. Then we have the pancreatic amylase, which is produced by the pancreas, and it is working in the small intestine, and where it breaks down again starch to maltose. Again we have maltase, which is produced by the small intestine, and it is also acting there itself, and it converts maltose to glucose. Again, lactase is the enzyme which is produced by the small intestine. The site of its action is also the small intestine, and it breaks down lactose to give rise to glucose and galactose. Similarly, we have the protein digestion carried out by enzyme pepsin, trypsin, peptidases. Nucleic acid digestion also is carried out by the enzyme nuclease, etc. The fat digestion is carried out by the enzyme lipase, which is very important enzyme produced by the pancreas, and the site of its action is the small intestine. So it converts the fat taken by all of us, consumed by us, to give rise to monoglycerides and fatty acids. So these are some of the digestive enzymes which you also students of biology would have learnt it in detail in their biology classes. Next, let us proceed towards the other functional aspect about proteins, the vitamins or vitamins. Now, what is important to the human body? Besides oxygen, which is mandatory essential component, water, another essential component for the human body. Other than that, we have fats, carbohydrates, proteins, and large number of organic compounds which are needed for our normal metabolic processes and for the life growth and the health of human beings and animals as well. There are certain other important compounds which are required in very small quantities, but they are performing very specific biological functions for our optimum growth and health, and also to sustain life. Such organic compounds are called vitamins. They are called vitamins because it was originally coined from the Latin word vita. Vita, V-I-T-A, means life, and vital plus amine was earlier identification of the organic compound because most of them had amino groups, NH2 groups. So they were all called vital plus amine. Vitamin is the word that has come up. Slowly, it has now been identified that not all of them will contain amino group. So therefore, the vital plus amine was changed to vitamin. That is how the word has come up. Now, when we are talking about the sources, so what are the sources for vitamins? They are milk, butter, cheese, fruits and green vegetables, meat, fish, eggs, etc. Commercially also, vitamins are available. They are available as tablets or capsules which are available in the market. So these are some of the important vitamins which we all are possessing and requiring. Vitamin A, B, C, D, E and K. They are very important vitamins for our body. What is important is most of the vitamins cannot be synthesized in our body. That is why we have to take it in the diet. From our diet only we get it. But what is important is, mercifully, plants can synthesize all of the vitamins. And very important, students, you must be amazed by this, that the bacteria which is present in the gut, it can also produce some of the vitamins which we require. But we cannot synthesize the vitamin in our body, but the bacteria in our gut can produce some of the vitamins. So therefore, as I said, all the vitamins are generally obtained from our diet. So we are designating the vitamins as I read out for all of you. Vitamin A, B, C, D, etc. And some of them have also got further subgroups like B1, B2, B6, B12, etc. Let us talk about the classification of vitamins. Vitamins are classified into two groups depending upon their solubility. They could be soluble in water or they could be soluble in fat. So whenever we are discussing the fat soluble vitamins, now these vitamins are soluble in fat and oil, therefore they are insoluble in water. Which are they? They are vitamins A, D, E and K. They all are fat soluble. There's a very popular question asked in a board exam as well. Give the example of fat soluble vitamins. Where are they stored? They are all stored in the liver and in the adipose tissue, because adipose tissue is the fat storing tissue in our body. Let us proceed towards the water soluble vitamins. So as the name itself tells, they are all soluble in water, like vitamin B and vitamin C. They are both soluble in water. Now, one important point which we all should not forget, that as these vitamins B and C are soluble in water, they are easily excreted along with the urine. Hence, they cannot be stored in our body only with an exception of vitamin B12. Therefore, it is very important that these two vitamins, vitamin B and vitamin C, are regularly supplied in our diet, as they are all flushed away along with the urine. They can't be retained in the body. Therefore, they need to be again taken in our diet continuously, only with the exception of vitamin B12. Let us now look into the different vitamins and the deficiency or the lack of vitamin which will cause certain diseases in us. So if you are talking about vitamin A, where do you get it from? Fish liver oil, carrots, milk, butter, etc. And the deficiency of vitamin A would cause night blindness. Okay, this is xerothalmia, which is hardening of the eye. Night blindness is caused by it. Vitamin B1, if is not sufficiently present in the body, it causes beriberi, a disease called beriberi. So what does this problem cause by beriberi? There would be retarded growth, loss of appetite. So how do I get vitamin B1? From milk, green vegetables, yeast, cereals, etc. Proceeding towards vitamin B12, vitamin B2, where we are having the problem or deficiency of vitamin B2 causing fissures at the corners of the mouth and the lips, digestive disorders, burning sensation of the skin. Always the skin seems to burn, giving us a burning sensation. And this all can be solved by taking vitamin B2 available from milk, egg white, the liver and the kidney of animals. The next one is vitamin B6. If there is a deficiency of vitamin B6, there would be convulsions caused in the brain. So it is a very, very hazardous problem. So therefore, we must replenish vitamin B6 in our body. And how do we get it from? Milk, yeast, egg yolk, cereals, grams, etc. All these will give vitamin B6. Vitamin B12, it is deficiency of it causes pernicious anemia. That is, there will be a hemoglobin deficient with RBC count, and this problem could also be sorted out by taking vitamin B12 regularly from eggs, curds, fish, meat, etc. The next vitamin is vitamin C, and if there's a deficiency of vitamin C, it causes scurvy, bleeding of gums. So gums will be bleeding even if you touch the gum. That is because of vitamin C deficiency. So how can I get vitamin C? From citrus fruits, from leafy vegetables, from amala, etc. Vitamin D is called sunshine vitamin, and vitamin D deficiency will cause this big problem like rickets, which is bone deformities in the children, osteomalacia, where the bones become very soft, joint pains would be there in adults. And how do you sort this out? It is by exposure to sunlight, taking fish and egg yolk. It's very interesting observation, children, that Indians lack vitamin D, and actually speaking, in our country there is no dearth of sunshine. We are having plenty of sunshine, yet in our body we are having this deficiency called vitamin D deficiency, the sunshine vitamin. Vitamin E, the deficiency of which will cause muscular weakness, fragility of RBC, it breaks down otherwise quickly, and this is sorted out by taking vitamin E from sunflower oil, vegetable oils, wheat germ oil, etc. Vitamin K, the deficiency of this will increase the blood clotting time. You know that the blood should clot within a specific period of time quickly. Otherwise, any person could bleed to death if the blood would not clot quickly. So, if there is a deficiency of vitamin K, it takes more time for the blood to clot, and this is a severe problem. This can be sorted out by taking green leafy vegetables so that they provide vitamin K. So students, please memorize and practice this very well, this table, because questions are asked from this table only. The deficiency disease caused by a particular vitamin and the name of the vitamin as well. So please do practice this very, very well. Now let us go towards the other functional aspect about protein, which are the hormones. The hormones in the human beings and other multicellular organisms are providing some kind of communication between the various cells to regulate the activity and the development of the cell. So this kind of communication, what is required, is only established by this class of compounds called hormones. So the hormones are generated or secreted by ductless glands called endocrine glands, and therefore they are called as the biochemical messengers. They are very important to all of us because they control the physiological activity such as growth and metabolism. Some of the major functions of hormone is to alter the rate of specific reactions of certain target cells. Let us understand the classification of hormones. Hormones are broadly classified into steroids and non-steroidal hormones. Steroids are further classified into corticoids and the sex hormones. The non-steroid hormones are classified into the peptide hormones like the insulin, the glucagon, etc., and amino acid derivatives, which is connected to the thyroid gland, the thyroid hormones, and other category called the miscellaneous cytokinins, etc. The sex hormones, what we are all studying about, are of two types: the female sex hormones and the male sex hormones. The female sex hormones are further classified into estrogen and progesterone. The male sex hormones are called testosterone. So this is a brief view about the classification of hormones. Let us talk about the steroid hormones. The steroid hormones are broadly classified into the corticoids and the sex hormones. When we are talking about the corticoids, where are they produced? They are produced by the adrenal glands, the cortex of the adrenal glands. And they are of two types: glucocorticoids and mineralocorticoids. Now, what does this glucocorticoids do? They are very important in the carbohydrate metabolism. They control it, and they also will modulate the inflammatory reactions which are caused in the body due to stress. The other one, mineralocorticoids, they will always control the quantity of water and salt that could be excreted by the kidney. When we talk about sex hormones, they are released by the gonads and they are of three types. Testosterone, and it is produced by the testes in the males. It is responsible for development of secondary male characteristics. Estradiol is produced in the ovaries of the females and it's responsible for the control of the menstrual cycle. Progesterone is also responsible for the preparing the uterus whenever the fertilized egg has to be implanted. So these are some of the functions of our sex hormones and our adrenal cortex hormones, the corticoids. Let us observe the picture of estradiol. This is the estradiol hormone and this is the testosterone hormone. If you are wanting to have a look at the progesterone hormone, this is the formula for progesterone hormone. Let us now proceed towards the non-steroids. The non-steroids are of two types, the peptide hormones and the amine hormones. Peptide hormones are basically containing the polypeptides, which are of two types, insulin and glucagon. Insulin, as you all know, are secreted by the beta cells of the pancreas, and it is the one which will maintain the blood sugar level, blood glucose level, and it will convert the excess of glucose present in the blood into the glycogen. So this is how the structure of insulin is in the what is generated or produced by the body. So we are talking about glucagon now. Glucagon is the one which will increase the glucose level in the body. So sometimes some people will have excess production of insulin, so excess of glucose is converted to glycogen and they all can suffer from the deficiency of glucose energy content in the blood, and as a result it will be a problem to them, which is sorted out by the body releasing the hormone glucagon. The next is the amine hormones. They are of two types. They are adrenaline, which are called as epinephrine and non-epinephrine. So some people also call it as epinephrine, norepinephrine. What do? They break down the glycogen present in the liver into glucose. The other hormone is the thyroxine hormone. It is always found in the thyroid gland. It's produced there. What does it do? It's a very important function. It maintains the growth and the development of the body. The structure of thyroxine is just presented before you. It's a very complex structure. Now children, let us proceed towards the other important components of our biomolecule, which are the nucleic acids. All of us are aware that every generation of species will resemble its ancestors in many ways. So, why is this happening? It has been established that the nucleus of the living cell is responsible for the transmission of these characteristics from one generation to another. So, what are these components responsible for this? They are all the chromosomes. And what are the chromosomes made up of? They are made up of nucleic acids. So, nucleic acids is the one which is responsible for the transmission of the hereditary characteristics from one generation to another. So what are these then? Nucleic acids are long chain polymers of nucleotides, and therefore they are all called polynucleotides. It is of two types. The deoxyribonucleic acid DNA, the structure is here, and ribonucleic acid RNA. So the difference between the two is only with reference to the sugar component present in the two molecules. When we are discussing the composition of nucleic acid, we understand that the nucleic acid is made up of many nucleotides, and each nucleotide has three parts. What are they? The pentose sugar, phosphoric acid, and the nitrogen containing heterocyclic base. So what is this pentose sugar? The pentose sugar are of two types. If it is observed in the DNA, the sugar moiety is beta D2 deoxyribose. So will you observe at carbon number 2 there is no OH group, it is deoxyribose which is found in the DNA. The other one is the ribose sugar which is present in the RNA. So here at carbon number 2 there is the oxygen present in the form of OH. If you compare it with deoxyribose at carbon number 2 there is no OH. That's the only difference between the two. The rest of the structures are similar. So the DNA has beta D2 deoxyribose, it's a beta configuration, and RNA has beta D ribose as carbon number 2 has the oxygen atom in the form of OH. The nitrogenous bases which are present in our nucleic acids are broadly classified into the purines and the pyrimidines. The purines include the bases adenine and guanine. They are abbreviated as A and G. These are the structures of adenine and guanine. You need not memorize the structures for the board examination. You should know them. That's all. The pyrimidines include cytosine, symbol C, thymine, T, and uracil, U. So these are the structures of cytosine, thymine, and uracil. You don't have to memorize the structures again. So let us understand again what are the bases present in the DNA. Adenine, guanine, cytosine, and thymine are present in the DNA. When you go towards the RNA, we have adenine, guanine, cytosine, and uracil. Yes, students, the common bases in the DNA and RNA are adenine, guanine, cytosine. The DNA is having thymine, whereas RNA has uracil. So let us remember this as DNA has A, G, C and T, whereas RNA has A, G, C and U. You must remember this very well. Let us understand the structure of the nucleic acid. The structure of the nucleic acid is started with the terminology nucleoside. What is this nucleoside consisting of? It has the attachment of a base at one dash position of the sugar. That means to the sugar moiety at position 1, the base is attached. So whenever we have the sugar combining with the base, you produce nucleoside. So I was talking about, this is the sugar moiety and we are having carbon number 1, 2, 3, 4, 5, which is all indicated with dash so that you can identify the position of the sugar. And at this carbon number 1 position of the sugar, what is attached? The base. What can that base be? The purines or pyrimidines. So therefore, this sugar moiety along with the base together is called as what? The nucleoside. Let us now understand a nucleotide. Whenever a nucleoside is linked to phosphoric acid at the 5' position of the sugar moiety, we get a nucleotide. So what is that? It is a very simple structure where the nucleoside is reacting with the phosphate group to produce a nucleotide. So let us take, this is the structure of our nucleoside. Again, I go back here. There's a nucleoside. At carbon number 5 of the sugar moiety, which is called the 5' position, you are going to have phosphoric acid group binding with it to form an ester linkage. And thus, the sugar at 1' position has the base and at 5' position has phosphoric acid group through an ester linkage, giving rise to a nucleotide. So a nucleoside plus phosphate group gives rise to a nucleotide. Now these nucleotides are joined together by a phosphodiester linkage, which is always between the 5' and the 3' carbon atoms of the pentose sugar. So the link is always between the 5' and the 3' carbon atoms of the pentose sugar. So if you want to understand the formation of a dinucleotide, so let us understand this is one nucleotide, sugar. The base is present at 1' position and at the 5' position of the sugar, phosphoric acid group in the form of phosphate is present. It reacts with another nucleotide. This is 1' position, the base attached to it. At the 5' position, the phosphate group is there. When these two combine with each other, they give rise to a phosphodiester linkage, which is indicated here, phosphodiester linkage, which is always between the 5' and the 3' position. So this is your 5' of other nucleotide and this is a 3' of the first nucleotide. So these two are linked together, 5' and 3' are linked together by the phosphodiester linkage. This is how a typical dinucleotide is formed. So in a simplified form to understand it, so every sugar will have a base and it will also be bonded to a phosphate. Again that phosphate is bonded to the sugar at the fifth position called the 5' end. So that's it. The sugar is bonded at 1' position to the base. The sugar is then also bonded to the phosphate group, and then this continuously goes on. So you are having the base, the sugar, the phosphate linkage again and again combining together to give rise to the nucleic acid molecule. So the polymer of ribonucleic acids or ribonucleotides are called as the RNA, the ribonucleic acids. So if you are talking about the polymer of deoxyribonucleotide, then it is called as deoxyribonucleic acid, the DNA. This is how you are producing both the ribonucleotides, RNA as well as the DNA. So what is the importance of these? The sequence of nucleotides in the chain of the nucleic acid is giving the primary structure of the nucleic acids. So the nucleic acids also have the secondary structure. Watson and Crick, the two scientists, eminent scientists, who gave us the double-stranded helical structure for the DNA. The structure of the DNA as presented by Watson and Crick model describes the DNA as it having two strands which run in opposite directions and are held by hydrogen bonds between the base pairs. So you can understand this is the one strand, it's running from 5' to the 3', and this is the other strand, the blue color which you can see, which runs from 3' to the 5'. So here the two strands are running in opposite directions. What happens here is the two strands are held together by hydrogen bonds, and the hydrogen bonds are between the respective bases. Adenine will form a bond with thymine, whereas cytosine will form a bond with guanine. This is how you are forming the bonding. It is noticed that adenine always has two hydrogen bonds with thymine, whereas cytosine will form three hydrogen bonds with guanine. So both adenine and thymine, cytosine and guanine, they are all linked to each other through hydrogen bonds. So hydrogen bonds play a very dominating role in the stability of the structure of the DNA. So students, henceforth, all of you must thank hydrogen bonding for these kinds of stability which is found in the molecule of DNA. The RNA in comparison is a single-stranded helix. So it doesn't have two strands like our DNA. The RNA molecules are of three types, and each type perform different functions. The RNA are named as messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA, it is the tRNA. They play a very important role in the protein synthesis. Now, what is very important for all of us is the differences between the DNA as well as the RNA. The DNA and RNA, as you can see here, can be differentiated as follows. The DNA has deoxyribose as the pentose sugar, whereas the RNA has ribose as the pentose sugar. The DNA contains thymine, it does not contain uracil, whereas RNA contains uracil, it does not contain thymine. DNA has a double-stranded helical structure. You can see this here, double-stranded helical structure. RNA has a single-stranded helical structure. It's a single strand. DNA is necessary for hereditary, the transmission of the hereditary characteristics, hence it is the chemical material of the hereditary. But RNA is not responsible for this transmission of characteristics through hereditary. DNA molecules cannot and do not synthesize proteins, but they only transfer the coded message for the synthesis of proteins in the cells, whereas the proteins are synthesized by the RNA. So if I want a protein to be synthesized in the body, it's only with the help of RNA. But the message for the synthesis of protein is stored in the DNA, which is transferred. So both work together for actually, in a nutshell, for the sustenance of life. When we proceed towards the biological functions of nucleic acid, we understand DNA is the chemical basis of hereditary and may be regarded as the reserve of the genetic information. And DNA is exclusively responsible for the maintenance of the identity of the different types of organisms, species of organisms for thousands and thousands of years. The DNA molecule is capable of self-replication and it can produce the daughter nuclei and transfer the genetic information. Thereby, it is capable of maintaining the identity of the different types of species on this planet. So students, with this we conclude the unit 14 for biomolecules. So I thoroughly enjoyed presenting the videos to all of you, and I'm sure all of you would also have understood and enjoyed the video sessions and learnt as well the chapter on biomolecules. So I'm signing off now. Thank you very much, and I want all of you to also take down important points and start preparing the lesson, the biomolecules, for your examinations. Thank you once again.